welcome.
So my name is Kevin Matthew, and this year I worked on analysis on the regulation and function of DNA polymerase and cystic saccharomyces conjugate. So in 2011, nearly 600,000 patients have died of cancer alone. And a large amount of that is due to rising skin cancer, which is largely caused by UV radiation. And a large amount is due to UV sunlight. And how UV radiation creates lesions within the DNA strands, as you see here. Now, these damaging agents will create lesions as black or cyclonutrient-mediated diamonds, which are the lesions inside the DNA. Now, when these lesions are created in the DNA strand, there are a few choices that the cell can choose to go with. Either it can undergo repair mechanisms, or if that is bypassed, it will undergo apoptosis or become mutagenic and become cancerous. So to prevent the option of becoming cancerous, these repair mechanisms are designed to actually bypass the lesion. And one of these repair mechanisms that I focused on was translation synthesis, which is coordinated by DNA polymerase A, or PAL-A. Now, how PAL-A works is that in this diagram over here, when the normal replicator machinery seen as the blue blob here reaches the lesion that is present in the DNA strand, it stops because it cannot overpass that DNA lesion. And since there is no basis there to correlate to determine which basis will go opposite of that lesion. So what happens is that polymerase A, seen over here with the red, will actually bypass the normal polymerase and actually take data or understand the basis from the opposite strand and actually match it along that lesion, and then isolate that lesion in just one cell when it replicates. So it does not undergo and it does not lead to apoptosis or cell death. So how we are determined to analyze the effect of polymerase A, we want to gain a greater knowledge of how polymerase A actually functions within the cell. We understand what polymerase A does, but we do not understand how it's regulated throughout the cell cycle and what actually initiates the presence of DNA polymerase A. So what our goal was to use S-POMB strains to actually determine the presence of polymerase A over multiple cell cycles in each phase of the cell cycle. So multiple generations over each phase of the cell cycle. So how we did this was through S-POMB strains using a lithium acetate transformation to actually plug in a YFP tag onto the polymerase A. Now this will allow us to actually do quantitative analysis and actually gain information through two main protocols, which is the Western blot protocol and the 96-well-K protocol, which I developed. So our first methodology would start with our derivative preparations. Now the point of the derivative preparations is to arrest the cell cycle at G1 and G2 phases. Now, by arresting at the G1, G2 phases by using certain mutations, this will allow us to use the Western blot protocol and determine the presence of polymerase A at one phase of the cell cycle with the G1 phase and the G2 phase. So this will give us knowledge of the presence of DNA polymerase A over these certain phases. Now, by force mating the cells along with these certain mutations and with other cells, we'll get the key strain that we need to use, and then we'll extract the DNA from that strain that we have acquired, and then do PCRs on the DNA that we acquired to determine if that is the exact sequence or if that's exactly where the polymerase A is supposed to be, and if that YFP tag is exactly on that polymerase A. So for preparative screening PCRs, we would then run it on gels and actually extract the bands, and then again send it to sequencing. And from that moment, when we understand that that is the exact place that we have that tag, we would go on with our Western blot protocol. Now, with the Western blot protocol, we will be able to analyze, as I mentioned before, the G1 and G2 phases of the cell cycle and the presence of polymerase A at that certain phases. So by determining the presence of that, we can also acknowledge the origins of polymerase A and how it actually functions within the cell. But the problem with the Western blot protocol is that it can be taken advantage of by actually either increasing the amount of antibodies or decreasing the amount of antibodies that is placed when we're proceeding with the procedure. Because by altering the presence of those antibodies, the amount of light that's emitted from when we're developing the film is going to vary. So there's a lot of human error that can be placed on exactly how much polymerase A is present at that level. So what we developed was the 96-well-K protocol, because this will allow us to actually view the cells, actually gain quantitative analysis of the cells of presence of polymerase A, at varying lengths and actually see the presence of polymerase eta through all parts of the cell cycle over multiple generations. So how we did this was through a 96-well-paired protocol, which would actually contain strains, actually have individual colonies and experiments in 96 wells. And because this will allow us to actually run 96 simultaneous experiments instead of having just one experiment over a 12-hour span. So having a we can save large amounts of time with the use of 96 well-played protocol and gain over 96 more sets of data points compared to just one experiment. So how does 96 well-played protocol that I developed, I use a poly-D-lysine polymer, which would actually keep the cells on a monolayer and actually prevent the cells from clumping, which would alter the data that we would get from analyzing the presence of polymerase eta by using a program that we have, so Velocity. So after we create the cells, after we have our cells that we've chosen that contain the YFP tag, we would first insert it and then use a low melt agarose that would have the agar at especially 30 degrees Celsius, which is the fit temperature for the cells. And then as we pour it, then we will centrifuge it and allow the cells to actually rest on the bottom of the glass and have the agar solidified. And then from that point, we would actually use a machine to actually irradiate the cells at various levels from 0 joules to 1500 joules of radiation to actually mock the presence of UV radiation on these cells. And after these are irradiated, we will then send it onto a microscope to run a live image over a 12-hour span. And then what you would see is from the 0 hours, well, the image that you see is the 0, 3, and 6, and 9 hours, that you can see how the cells actually divide and replicate. And then from this data, we can actually create regions of interest on the nuclei itself that contains polymerase eta and determine the presence of pixels and the amount of fluorescence as it goes through the cell cycle. So when our results based on our Western blot protocol, at first it was initially difficult to actually find and actually determine the presence of polymerase eta, indicating that there is low levels of polymerase eta. And without the use of the UV radiation, it shows that it's possible that UV radiation is a large key to actually triggering the presence of polymerase eta. Now, the second part, which is what we're working on, is through the analysis of the live videos that we obtained. We have not yet analyzed the videos, but based on our live image of the videos, you can see that the polymerase eta, or the amount of fluorescence, doesn't actually alter, which would support our data from the G1 and G2 from the Western blot protocol, which says that G1 and G2 phase there really isn't a variation in the presence of polymerase eta. But we cannot really determine that because our eyes can be susceptible to different variation. We cannot really understand if there is any slight variation. So understanding by using regions of interest with our program, we can determine there is slight variation throughout the cell cycle. So based on our preliminary results, we can see that G1 and G2, based on our preliminary results, you can see that the polymerase eta is actually present throughout the cell cycle, which can determine that there really isn't that Pol eta protein turnover that was observed before, or which is triggered. So our future research is to actually gain more knowledge by actually gaining the knowledge of the polymerase eta and actually the presence of polymerase eta throughout the cell cycle. And from that knowledge that we gain, we can also probably determine the origins of how cancer actually affects the cell and actually prevent the cell from actually taking the options of undergoing apoptosis or becoming mutagenic, and actually somehow initiate the cell to force it to a repair mechanism and actually prevent these issues.